We're now going to move into the next segment of the course, Understanding and Measuring Health. And we're going to look at this by looking at the two core disciplines of public health, epidemiology and biostatistics. As I mentioned in the Intro to Public Health slide, epidemiology is the science of public health, and it's the study of patterns of diseases and injury and human behaviors in human population, and then taking that information through research to really understand what the root causes of the disease are so that we can plan for prevention and control of those health conditions. And epidemiology and epidemiologists work hand in hand with biostatisticians. And biostatisticians use mathematical modeling of the data collected often by epidemiologists to come up with risk estimates, to be able to say what proportion of the population has what disease or what behavior, so that we can then look to see where our efforts should be spent and what we should be doing, what works and what doesn't work. And so the first epidemiologists really were about recording the observations of the world around them. They were often physicians who would see either new or emerging diseases and try to understand what the root causes were. And so epidemiologists today continue to look at these patterns of disease occurrence, try to see what the etiology or the root cause of those diseases are, and then use that information to recommend means of prevention or disease control. And they deal with diseases that are endemic, epidemic, or pandemic. And what do I mean by these terms? Well, I'll get to that in just a moment. Epidemiology at its core, like public health as a field in general, is multidisciplinary. And so it requires input from a lot of different disciplines to generate the hypotheses that we work with. And this includes individuals in the medical and biological sciences, the social and behavioral sciences, and as I said, biostatisticians who we work with very closely. And most epidemiologists also have a strong foundation in biostatistics. Epidemiologists often describe themselves in one of three ways: either exposure specific, so they might describe themselves as a nutritional epidemiologist. I'm interested in the way that food and patterns of eating and the types of foods we eat influence a multitude of different outcomes. They might describe themselves in terms of a disease-specific outcome. So I'm a cancer epidemiologist or a cardiovascular, and you're interested in all aspects of cancer and all of the risk factors that lead to the development of cancer or different cancers. And some people are very hyper-specific and say I'm a breast cancer epidemiologist, etc. And another big part of epidemiology are health services research and program evaluation, which is a big component of the program here at Montclair State University. And it's really applying the theories and the tools of epidemiology to look at programs and policies to see how they're working or to predict how they will work in order to generate new ideas. And so some terminologies that we use to describe disease occurrence, and these tend to be used mainly around infectious diseases, but can also be used around chronic diseases as well. The first is endemic. A disease that is endemic is something that you expect to find. There are certain diseases in certain geographic areas, and you expect to see a certain number of cases. So for example, in the United States, we have cold and flu season. So if you think about this area in and around New Jersey, typically we think of cold and flu season as running from November to April. And with that, we expect, as epidemiologists and those in the health field, that there will be a certain number of cases of flu each year. An epidemic is when the rate of a disease is higher than that which is expected. So there are seasons where we have different flu epidemics, where the rates are higher than what we would think, given what we typically know. An epidemic, which is also often referred to as an outbreak, can also be a case of a disease that you don't expect to see at all. So over the past few years, there was an outbreak or an epidemic of Ebola. And in the United States, we don't expect to see any cases of Ebola. So when you see 1, 2, 3, 4 cases, that's considered an epidemic or an outbreak. So again, it's the widespread occurrence of disease in a community or a geographic location at a particular time. And widespread can really mean anything more than what we typically expect. The difference between an epidemic and a pandemic is an epidemic that has spread beyond geographic and national borders. And so one of the last big epidemics would have been pandemics would probably be swine flu about 8 years ago. And it was determined to be a pandemic when it was epidemic in multiple countries that were not geographically connected and was spreading. But other things have been described as either an epidemic or pandemic. Things like obesity, which was initially described as epidemic in the United States, but is now spread to other parts of the world and is considered to be pandemic as well. And when we think about disease, we don't just think about the raw numbers, how many people got the disease, but really we look at it as rates of disease occurrence. And there's two ways to consider this. One is prevalence, which are the number of cases in a population at a specific time. And incidence is the number of new cases in that population at a specific time. And an attack rate is really the number of cases in a population divided by the population at risk. So we'll talk about that a little bit more in a minute. But another way of thinking about these measures of morbidity or illness, prevalence is the number of existing cases, whether they are new cases or they're cases that people were diagnosed the year before and continue to be sick because they have not recovered, divided by the population at risk, while the incidents are the number of new cases in a given time period. So when we think about things like heart disease, the prevalence would be everybody currently living in the United States who've been diagnosed with heart disease divided by the total population in the US. So they could have been diagnosed 5 years ago, 10 years ago, this year, it does not matter. The incident would be how many cases of heart disease were diagnosed this year divided by the total population. And so they're slightly different, but those are important differences when we talk about incidence and prevalence. Because a disease can have a high incidence and a low prevalence, meaning people can get sick and then recover. So the incidence is high, but there aren't a lot of old cases, or there can be high incidence because there are a lot of new cases, but they die. And a disease can also have a high prevalence with a low incidence. So there aren't a lot of new cases that are diagnosed each year, but the people are diagnosed and they live for a long time. And so there's this cumulative impact where people are living for a long time. And you can have diseases that have both high prevalence and high incidence. And so let's consider we have a new disease, disease X. And we have a population of 100 students, of whom 25 contract this new disease. What would the incidence rate be? Well, it's a new disease. We haven't seen it before. So our population at risk is 100. That'll be our denominator. And the number of new cases would be the numerator, or 25. So in this case, 25 divided by 100, so about 25%. And we tend to multiply by either 100 or 1,000 because we like working with whole numbers. So in this case, the incidence rate was 25%. The prevalence rate would likewise be 25. There weren't any old cases. These are all new cases. So we have a prevalence of 25%. Now let's assume 15 people die from the disease, 5 remain stricken, and 5 recover. So what is the mortality rate? Well, in this case, the mortality rate would be 15. So 15 people died out of the total population of 100. So we have a 15% mortality rate due to disease X. But we have a different measure called the case fatality. And in this case, the case fatality looks at how many people died divided by the number of people who were stricken, the number of people who had the disease. So in this case, the case fatality rate is 60%. That's much higher. And the difference between these two are you can have a disease that has a relatively low prevalence, so not a lot of people are contracting it, and therefore your mortality rate would look lower, 15%. But it could be a highly fatal disease. So not a lot of people are getting it, but the people who are getting it are at very high risk for dying, and therefore have a high case fatality rate. You weren't supposed to see that. This came in wrong. So assume 1 year later, there are now 10 new cases in addition to the 15 people who died from the disease, 5 who recover, and the 5 who remain stricken. So now what's the incidence rate? So the incidence rate in this case is going to be 10 out of 85. So our population declined by 15. Those are the 15 who died. And we have 10 new cases. So this year, we now have an incidence rate of 11.8. The prevalence rate will be how many people survived. So we have 10 new cases, plus the 5 who remained stricken from last year, 15 out of 85, and our prevalence rate is now down to 17.6%. I'll give you a minute just to think about that. And of course, as with anything, if you have any questions, you can always either contact me through the Canvas email or better yet, post it in the discussion board, and everyone can benefit from your question and my attempting to answer it. So looking at the difference between mortality rate and case fatality rate a different way, your mortality rate, the numerator is the number of people who died during a certain period of time with that disease over the entire population, whereas the case fatality are the number of people who die of a specific disease during a certain period of time, divided by the number of people with that disease in that population. And so let's take another look, or a closer look, at infectious disease epidemiology. And infectious disease epidemiology is really, or what's often now called emerging infectious diseases, was really the historical basis of epidemiology. In the early, in the 1800s, the 1900s, people were largely not dying of things like heart disease and cancer, diseases that you live with for a long time. They were dying of things like dysentery and cholera. And speaking of cholera, we have here John Snow, who is considered to be the father of modern epidemiology. And John Snow observed that cholera epidemics followed major routes of commerce and warfare, and that cholera almost always appeared first in the seaports. And I will give you a very brief history of epidemiology now. So John Snow looked at these cholera epidemics or outbreaks and noted a few things. He determined that this disease could be communicated through person-to-person transmission and must be caused by some material that passed from the sick to the healthy and had the property of multiplying in the systems of people it attacked. And he came to this conclusion by looking at patients and where they came from, so trying to track those patterns of disease. So he noted that cholera could be communicated from sick people to healthy people, but he also noted that people who were caring for cholera patients did not always develop cholera. He also noted that close contact with a cholera patient was not necessary to become afflicted. It was sufficient in some cases, but not always necessary. And he noted that one of the highly afflicted populations were coal miners, and these were individuals who were working down in the coal mines. They did not have bathrooms. They did not have places to wash their hands. So if they were going to bathroom in the coal mines, they were then eating their lunch in those same coal mines with their unwashed hands. And he noted that while cholera seemed to be concentrated in these poorer neighborhoods that were very overcrowded, it could reach beyond them as well. And so he had the question of how is cholera affecting so many different people of so many different classes? And his solution was that there must be some sort of, again, common vehicle. He determined that because it was a gastrointestinal disease, cholera causes severe diarrhea, and you typically die from dehydration as a result. And he thought the solution could be water, which is something that was used both for drinking and culinary purposes. And so one of the first things he did was get a map of London, and this is a map of part of London, and he mapped the places of residents who died from cholera. So these bars that you see, some are shorter, here maybe one individual, 2 individuals, 3 individuals. In other cases, there were a number of individuals living in a particular either residence or neighborhood that were dying. And he mapped those in relation to where the water pumps are. So here's the Broad Street pump. There's a pump over here near Church Street. I think those are the 3 that are shown on this map. And he determined that cholera epidemics could be related to the nature of the water supply. So different districts in London had different rates. And so you can see over here by the Broad Street pump, there seemed to be more cases. And he noted that mortality or death rates were impacted by poverty, crowding, and the want of cleanliness. And so in another map, and this is John Snow's, a replica of his actual map, or a picture of it, he outlined the different areas. So the blue, which turned to green because it is faded, maps where the Southwark and Vauxhall Company provided water, and in the red area, the Lambeth Company, which is outlined in red. And so he was looking to see where there are differences. If you want to learn more about this, here's a website you can check out for those budding epidemiologists amongst us. So the grand experiment of 1854 involved two different water companies. Between 1845 and 1852, both companies supplied water to people living in the same area with polluted water that was drawn from the River Thames. Then in 1852, the Lambeth Waterworks Company decided to move its water intake upriver to a cleaner location, while the Southwark and Vauxhall Company left its intake at the same contaminated location. When the next cholera epidemic appeared in 1853 to 1854, some neighbors were unknowingly receiving cleaner water from the Lambeth Company, while others consumed more polluted water from Southwark and Vauxhall. And this change in the location of water intake was the basis for a natural experiment. Dr. Snow did not change the water intake. This is something that happened, and he was merely observing these patterns. And this allowed Dr. Snow to compare mortality patterns by water source and strengthen his hypothesis regarding the transmission of cholera. And so this is what he found. He looked at the number of houses that were served by Southwark and Vauxhall compared to Lambeth, and the number of deaths from cholera. And he then divided these two and found that for homes served by Southwark and Vauxhall, there were 315 deaths from cholera per 10,000 homes, versus only 37 deaths per 10,000 homes in the Lambeth Company. And how was the epidemic stopped? Well, by plotting the cholera death on a spot map, John Snow identified the Broad Street pump, a replica of which you see here, as the source of the cholera outbreak in 1854. He famously removed the pump handle and the number of deaths began to abate. Now, there are questions whether a number of deaths would have begun to abate before the removal of the pump, but nonetheless, this was the first sort of noted case of somebody plotting the patterns of a disease, developing a hypothesis, analyzing the data, and then acting on it to try to prevent or control. In the background of this picture, you also see the John Snow pub, which you can all visit if you decide to go to London. When I went to London 4 years ago, you see my picture of the pump. We call it the epidemiologist pilgrimage. And there is my son, much younger there than he is now, happily posing with the pump. And if you do ever go to London, you can go to the John Snow pub. The original pump has been removed. It was in the middle of the street. That's not practical in modern times, but it has been replaced with this lovely red brick, which you can find and take a picture of. And so you can see that that took place there in London. It's not easy to find, and most people in London are not very helpful in finding it. I will warn you. And so what were some of the outcomes? Well, he recommended that strict cleanliness should be used when dealing with the sick. So if you're dealing with someone with cholera, you should wash your hands. That soiled bed linens should be immediately immersed in water. So again, these individuals were dying of diarrhea, uncontrollable. They were soiling the bed linens. And so those should not just be left out and about. Shockingly enough, he thought that water for drinking or food preparation should not be contaminated with sewage contents. And he recommended to the government that they not disguise the facts about cholera from the public, that they should be honest. So in some ways, this was also the beginnings of policies around health communication. He recommended that we be careful and wash our hands thoroughly, and that particularly for low-income communities where there was a lot of overcrowding and therefore a higher rate of disease transmission, that the government should really provide adequate housing for the impoverished classes. And so that was the beginning of epidemiology. And as a result of some of that beginning work, and also work in the biological sciences and microbiology, and the advent of understanding of microbes, we've seen a significant shift in the leading causes of death. So in 1900, three of the top 5 leading causes of death were infectious. So we had pneumonia and influenza, tuberculosis, diarrhea and enteritis, and you also had things like diphtheria, another infectious disease. Whereas in 2015, this has changed. While pneumonia and influenza is still in the top 10, it's dropped down to number 8. And you see an increase in things like heart disease and cancer. People are living longer and therefore have time to develop these chronic diseases, which take time to develop. And so this is a quote that I love to share. The Surgeon General said, "Because infectious diseases have largely been controlled in the United States, we can now close the book on infectious diseases. Not something we need to worry about anymore." Well, that was said in 1967, and as we all know, the book on infectious diseases has certainly not been closed. If you look at crude infectious disease mortality rates from 1990 through 1996, what you can see is that the first dramatic decrease was brought about by having health departments in at least 40 of the states. You see a spike in 1918, which is a dramatic spike, which is due largely to the 1918 influenza pandemic. This killed millions of people. It started on the East Coast and spread across. So again, started at the seaports like many infectious diseases. In around 1910, you saw the first continuous municipal use of chlorine in the water. So again, there was a decrease in infectious disease rates because we had better access to clean water. And another sort of big dramatic drop was with the first use of penicillin used to treat bacterial infections. So again, the advent of antibiotics. There was another small dip when Salk introduced the vaccine for polio in the 1950s. But interestingly enough, what you see here, this is when, infectious diseases are gone. We don't need to worry about it. You do see this increase. Now, obviously, compared to the overall trajectory, that's not huge, but it is significant. And so looking at this further out, you see there's this sort of bump post-1980, which has come down slightly and again leveled out. So what was the reason for that bump in the 1980s? Well, it was largely the discovery of HIV and AIDS. Got away from me. And so if you think about this, focusing on people, okay, I'll come back to that thought in a minute. But you see this dramatic rise in HIV/AIDS. Once it is more recognized and we start developing treatments, this is when we develop the cocktail and people understand prevention, the rates have sort of declined and come down because HIV went from being an acute infectious disease where you die fairly rapidly to now become more of a chronic condition with treatments. But HIV isn't the only emerging infectious disease since 1967. We've seen repeated outbreaks of Ebola. There was Legionnaire's disease in 1976. And we've also seen things like West Nile virus, anthrax, SARS, the bird flu, and as few as 8 years ago, the swine flu. So these infectious diseases are not going away. How we prevent and control them still continue to matter. And an example of that is HIV. So if you look only at individuals who are 25 to 44 years old, it emphasizes the importance of HIV among the causes of death. Compared with rates of individuals at other ages, the rate of death due to HIV, is relatively high in this age group. But rates of death to the other causes are also relatively low. The disease was the leading cause of death amongst people 25 to 44 in 1994 and 1995. And in 1995, it caused about 32,000 deaths, or 20% of deaths, and of all deaths for that age group. And it fell from 5th place to 6th place through 2004. Within 2004, it causing about 6,000 deaths, or only 5% in this age group. However, HIV continues to remain among the 10 leading causes of death among people 25 to 44, particularly among particular groups. And so when we're studying infectious disease and thinking of it in terms of epidemiology, there are some concepts we need to understand, and we'll go through each of these. One is the relationship between the agent, host, and environment. If we're going to study a disease and its patterns, we need to know its modes of transportation. We need to understand the incubation period, the spectrum of disease, as well as concepts in herd immunity. And I'll go through each of these. So the theory of disease occurrence says that if the agent, in this case, people, humans, that's what we're studying in public health. The disease agent, the host, and the environment are all in balance, then disease won't occur. If you have high exposure to disease agents, so high exposure of virus or bacteria or certain fungus, that's going to throw off that equilibrium and you're more likely to get sick. If the host, humans in this case, have low immunity, so when you're initially born, if you are immunocompromised due to other conditions, if it's finals time and you're not sleeping well or eating as well, or if you're drinking a lot, that can reduce your immune system. You're more susceptible to becoming ill. And certain environment conditions can benefit the agent over the host. So in the summertime, it gets warm. There's lots of standing water that allows for the multiplication and reproduction of mosquito and the larvae in those standing water. So we get a higher population of mosquitoes. That increases the probability that those mosquitoes will carry West Nile. It also increases the probability that you will be bitten by a mosquito, which increases your risk of West Nile. So all of those things need to be in balance. And when they aren't, disease can occur. So what are some infectious disease agents? Well, things that we hear about a lot, viruses and bacteria. So the cold and flu are caused by viruses. Bacteria would be things like strep throat. It's important to note that those two things are very different. So bacteria are microscopic single-celled organisms. Viruses are DNA with a protein coating, and we treat them differently. So if you have the cold or the flu, you don't need antibiotics. What you need is an antiviral. And for the flu, there is now a good antiviral called Tamiflu. If you have strep throat, you need an antibiotic because that is a bacterial infection. But there are also fun things out there like prions, which are basically proteins that reside on the surface of brain cells and can cause things like mad cow disease. There are also certain fungus that can cause infection, so athlete's foot being a common one. And also things like protozoa and metazoa, which are either single-cell or multi-celled organisms that generally are parasites. So things like different worms that get into your system. And so when we think of the epidemiologic triad concepts, we think of things like infectivity. Infectivity is the ability of a disease agent to invade the host. So how many people are susceptible, how many people came in contact with the disease, and how many were infected. And this is tested in a number of different ways. The common cold has been, the infectivity of different strains of the common cold have been looked at by quite literally putting the common cold into people's noses and see who gets infected and who doesn't. Yes, people actually sign up for those studies. Hard to believe, but true. And we'll talk about that more later in the course. There's also something called pathogenicity. So of the people who are infected, how many show symptoms of the disease? You can be infected and never show symptoms. That's called an inapparent infection. And then you have virulence. So of the people who have the disease, how many die? So, pardon me again, sorry. We have infectivity, pathogenicity, and virulence. So the ability to infect, the ability to cause symptoms, and then the ability to kill the host. And all of those are going to be dependent on the condition of the host. So have you been exposed to this disease before and therefore have some immunity? Have you been vaccinated? How well nourished are you? Individuals who live where there is frank starvation typically don't die of starving, they die of infection because they're susceptible to those infections. Lack of sleep, you may find during finals time and you're studying and you're not sleeping as well, during finals or immediately after, you get sick because your body is worn down. And of course, poor hygiene, very important. Diseases are also classified by how they're transmitted. And so just like Jon Snow had to determine what was causing it, was this person to person, was it a common vehicle? Knowing the modes of transmission can help you understand the disease, find the root cause, and then ideally treat and prevent it. So you can have person-to-person contact. This can be respiratory, where you're coughing and you're breathing in or sneezing and not covering your mouth properly. Orogenital, so oral contact with genitalia, or skin to skin. Some examples are things like HIV and measles. You can have vectors, so through animals or other insects, so things like rabies and yellow fever. Common vehicle, cholera is spread through food and water, but so are things like salmonella, largely associated with chicken and eggs. And then mechanical vectors, so sharing toothbrushes or hairbrushes or someone's towel, but also things like doorknobs. So somebody sneezes, they sneeze into their hand, they touch the doorknob, you touch the doorknob, and now you touch your face, and you have their germs. And so different ways of looking at this classification by mode of transmission. So spread by a common vehicle, again, ingestion, inhalation, inoculation. Propagation by serial transfer from host to host. So this can be respiratory, anal, oral, or genital. When I say anal, oral, people get giggly in class typically. But what this really often means is somebody using the restroom, not washing their hand thoroughly, and then preparing your food and having fecal matter on your food, which you then ingest. There are certain principal reservoirs of infection. So humans, other vertebrates, other free-living infections. So there are certain infections that are innate that we carry with us and then can spread and don't necessarily kill us, but can infect other animals. We can look at the mode of transmission by how it enters or exits the human host. So does it impact your upper or lower respiratory tract, your gastrointestinal tract, your genitourinary tract, conjunctiva, which are your eyes, your skin? So is it getting into your skin through, you're swimming in tainted water and it literally gets directly into your skin, like leptospirosis, which comes from animals urinating in lakes and streams that you then bathe in. And that's actually been a significant problem this spring and has been impacting a lot of pets who people let their pets run into these streams. They drink the water, it gets into their skin, and it gets passed along, and they've been getting sick. It's been a big problem. Or things like percutaneous, where you're getting injected, whether it be through a needle or through mosquitoes, and then getting into your bloodstream. Do I have this one in here twice? Oh, so, sorry. There we go, got to the last one. And then there's how it cycles. So is it spread from man to man to insect to man, vertebrate to an insect, to another vertebrate to man? And so all of those things you really need to understand. And when I mentioned earlier about how epidemiologists work closely with people in other disciplines, this is where you're either going to want to have a solid founding or a grounding in the biological or chemical sciences, or work with other people who do. Another thing to think about are the incubation period. So you don't come in contact with an infection and suddenly show symptoms that same moment. There is a certain period of time called latency, which is the time between contact and the onset of clinical illness. And that's a time that's required for multiplication of that organism in the host to get to a point where the population is large enough to produce symptoms. And each infectious disease has its own characteristic period. And that's dependent on the rate of growth of that organism in the host and things like the dosage of the infectious agent. How much were you exposed to? How did it get in? So certain microorganisms, if they get into your lungs, they're going to spread much quicker than if you're just touching them. So the difference between touching a doorknob and then touching your eyes versus if you have young children, having them, sneeze directly in your face. I have 2 children. They have both done it. They're lucky. They're cute. The immune response of the host, as I mentioned just a moment ago. And because of the interplay between these factors, the incubation period is not the same for anybody. There's a range that we look at. But if we know the general range, if we know that, the cold exposure can take between 7 to 10 days, if we see an increase in the rate of people with the flu, we'll know, okay, let's find the index case and we can look back. How many people have they been in contact with over the past 7 to 10 days? So knowing that incubation period is really important and understanding and mapping the patterns of disease. And because epidemiologists and people in public health like triangles, I have another triangle for you. So the stages of disease. As I said when we talked about infectivity, not everybody who's exposed to a disease is going to become infected. So 90 to 95% of the infectious disease agents you come in contact are subclinical disease, either exposure without infection or inapparent infection, or infection without clinical illness. You're infected, your body fights it off, you never show signs, you move on with your day. Vast majority of the time this is the case. And about 4 to 8% of exposures to infectious agents, you show signs. You have clinical symptoms of the disease. Most of that is moderate to mild or mild to moderate severity. Some will present with critical to severe disease, and only 1 to 2% of all infections you come in contact with, and there's millions in your lifetime, will cause death. So the good news is the chance of you dying from an infectious disease, as I said, is relatively low compared to other things. And so there's this spectrum of disease, right? 90% of people who are exposed to measles will exhibit clinical symptoms. Let me clarify. 90% of people exposed to measles who are not vaccinated will show clinical symptoms, versus only 66% of mumps cases and less than 10% of polio cases. However, you don't want to be infected with any of them because they can all cause significant problems with the measles, particularly for women who are pregnant or of childbearing age. You can cause significant problems to the fetus. For mumps, it's very uncomfortable and can be particularly uncomfortable for men, as this tends to attract your testicles. And for polio, it can cause significant disability and death. And so while most people who are exposed to polio would not show clinical symptoms, those that do are going to have significant negative consequences. And so one of my favorite concepts in infectious disease epidemiology is this idea of herd immunity. So herd immunity came about through farmers trying to understand how much of their livestock needs to be vaccinated in order to prevent disease from spreading. Right? So it's very difficult to vaccinate every single one, but we don't want an epidemic or an outbreak of some disease that kill off the whole, your entire herd, and then you lose a lot of money. And so herd immunity is basically this idea that there's a decreased probability that a group will develop an epidemic if a certain proportion of immune individuals are in that population. So you just need just enough to reduce the chance of contact between infected and susceptible people. So in other words, the entire population doesn't need to be immunized to prevent the occurrence of an epidemic. And this theory or this idea was used to eradicate smallpox worldwide. So there was no way to vaccinate every single living person against smallpox. But if you were able to vaccinate enough people that those who were infected could not or reduce the probability of them coming in contact with people who are susceptible, eventually the disease would die out and you would not see any new cases. And that has been the case with smallpox. And things like polio, at least in the United States, have been eradicated. And so, how does this work? So let's imagine here's a population, there are 90 individuals. The immunity level is 32%, so not very many people have been vaccinated. That leaves 68% of the population who are susceptible. So you have your little index case over here, they start spreading the disease. And the individuals in the darker boxes are those who are susceptible. And because there are so many people who are susceptible, the disease spreads quickly. They're more likely to come in contact. And so, in this example, 55% of individuals will contract the disease overall, and of those who are susceptible, 84% will get the disease. There will be a few who don't. By chance, that could be their own immune response. Again, those host factors, but it's going to spread quickly. Let's look at the same population. However, let's change the immunity level. Now we have 85% immunity, only 15% susceptible. So now, instead of having 55% of the overall population contracting the illness, you have 4%. That's much lower. And of those who are susceptible, instead of 84% contracting the illness, you have 28%. And because those individuals aren't going to come in contact with other individuals as easily, we can control the disease and we can reduce the probability of there being an epidemic or an outbreak that's widespread. And so when we think about these diseases, we think about things like, again, the incidence, the attack rate, the mortality, and the case fatality. So the attack rate is this cumulative incidence in a group over a period during an epidemic. So in a given population, how many got sick? And so looking at this a little bit closer, we can calculate, let's say there is an outbreak of a foodborne illness, and we're trying to figure out what it was. And so what you would want to do is find out, okay, how many people ate this particular food that we think caused it? How many people didn't? And of each, who were fine, didn't get sick, and who did get sick? And then you can calculate your attack rate for each of those foods. Right? So the number of sick divided by the total, multiplied by 100 to get a nice round number during a period of time. So the attack rate for people who ate the food was 10 out of 13, or 77%. The attack rate for people who did not eat the food were 7 divided by 11, or 64%. Now you look at those and you go, well, a lot of people in each group got sick. I mean, is there really a difference between 77 and 64? There are two ways to determine this. One, you can look at the attack rate difference, sort of just, is there a difference in those groups? So 77 minus 64, there's a 13% difference. It looks like that's an increased risk. Or what you can do is get your attack rate ratio. So you look at the attack rate amongst people who are exposed divided by the attack rate of people who are not exposed. So in this case, that would be 77 divided by 64, or 1.2. If there is no difference, you expect that number to be 1 or very, very, very close to 1. In this case, it's 1.2. And so you could say that there is a 1.2-fold increased risk, or you can take the 0.2 and say there's a 20% increased risk of having the disease if you ate that particular food. So let's look at this a little bit more. There was this case, we tend to call it the epidemiologist picnic. So it's the summer, the Department of Health was notified that 6 people had come into the emergency room complaining of these gastrointestinal symptoms: vomiting, diarrhea, abdominal cramps. Everybody had eaten food from a local fundraiser at approximately noon. And we know that this fundraiser had served food from 10 a.m. to 6 p.m. At a local community center, and had delivered plates to businesses and other personal residences. So what do you need to know to decide, is this an outbreak? Is it an epidemic? Well, as we said, an epidemic is a number of cases beyond which you would normally expect. So do we typically expect 6 people to come into the emergency department or to the emergency room on any given August day complaining of these symptoms? If this is more than you would normally expect, then you can say there's something here we need to explore a little bit more. So let's look a little deeper. So the first important thing is you need to define what it means to be a case. So as public health practitioners and health department workers, we're going to define a case as symptoms of nausea, vomiting, diarrhea, cramps, and or abdominal cramps and or diarrhea within 24 hours of eating food from the fundraiser. We know that about 100 people attended the fundraiser. We were able to collect information from 75, and as it turns out, more than those 6 met the case definition. 6 presented to the emergency room, but 46% of people showed those signs and symptoms. And so, how would you calculate the attack rate? So overall, the attack rate would be, in this case, 46 people out of the 75 that we were able to collect information on. So there's an attack rate of about 61.3%. That seems high. We should look deeper. And so this is an actual line listing of an outbreak of gastroenteritis that happened in New York in 1940. And what they did is they have the subject ID, their age, their gender, the time of the meal, whether they were sick or not. If they were, when did the symptoms start showing, what time and what day. And then they asked them about all the different foods that were served. So we have baked ham, spinach, mashed potatoes, cabbage salad, jello, etc. Did you eat these things, yes or no? Now this seems sort of overwhelming, but from there what we can do is calculate who ate what and when, and trying to figure it out. So 14 different foods were served. Was it all of them? Was it one of them that caused the enteritis? And so in addition to calculating the overall attack rate, you can calculate the attack rate for each of those individual food items, for each group, exposed and unexposed, who got sick. So investigators put together this table. You have the number of people who ate a specific food, people who did not eat that food, who got sick, and the total number of people. So you'll see these totals are going to be different because not everybody ate every food. And then you can simply calculate the attack rate. Again, number of sick divided by the total for each food. So for the hamburger, 24 out of 46 people who ate the food got sick. So that's 52, an attack rate percent of 52%. And for those who did not eat the food, ate the hamburger, 15 got sick. So 51.7%. Again, it doesn't seem too different. And what you would do is you would calculate that attack rate for every single food item for people who ate it and people who didn't. So it seems over dividing. So far, not so bad. From there, what you need to look at is, are the differences amongst the people who ate the foods different than the attack rate for the people who didn't? So we expand our table by 2 columns. We can again look at that difference, just subtracting, and we can look at the attack rate ratio. So the attack rate different is just the attack rate in the exposed minus the attack rate in the unexposed. So the difference in the hamburger would be 52 minus 51.7, a difference of 0.03. Not very big. If there was no association whatsoever, it would be 0. That's pretty close to 0 in this case. And so you can calculate these. Now these have a negative, so it might be that eating the spinach salad or potato chips or rolls were protective. Or maybe that's all they ate. Potato salad, however, there's a pretty big difference there, 29.3. Same thing with the water, so there might be something there. Another way to look at it would be the attack rate ratio. Again, we're dividing instead of subtracting now. So you're dividing the exposed by the unexposed. So for the hamburger, it's a difference of 1.01. That means there's a 1% increased risk. That's not very big. Same thing for the hot dogs. But if you look here, 1.68, that's pretty big. That's the equivalent of a 68% increased risk. So, which food has the greatest difference in the attack rate? That's likely the food that is causing the outbreak. So you have your attack rate ratio, again, the potato salad, 1.68, 68% increased risk of enteritis if you ate the potato salad that day, which means that's most likely the cause. And so it's applying these types of things, looking at the incidence rate, looking at the prevalence, looking at the attack rate and the mortality rate, which really allows us, through the use of both the methods of epidemiology and the math of the biostatistics, to model and estimate the risk so that we can predict and we can make recommendations. So, as we say in the epidemiologist picnic, if you're going to a picnic in the summer, don't eat the potato salad. But the reality is, why might the potato salad be the case? So now you can look into that a little bit closer. Was it kept at the proper temperature? If not, that may have provided an ideal environment for the agent to grow, increasing the host exposure to a higher amount of that disease agent, which then allows a higher infectivity and pathogenicity. And so it all ties together. In the next video, we'll be presenting another case and we'll walk through a case together of another outbreak. So I look forward to talking to you more about epidemiology then.